Hey guys, so you can actually see me in this one, and that's because I'm going to be using some manipulatives that we're going to be talking about as we go through. So you definitely need to make sure you're kind of watching this as well. So last time we talked about proteins. It was one of our major macromolecules. Make sure that you're writing this information on page 16. Make sure you label it topic 13 enzymes, because that's what we're going to be learning about. So we talked about proteins, and I said that we're not really going to talk about them in too much detail in that particular lecture. Well, this is the lecture that we're going to be focusing on proteins. Because proteins are, or rather enzymes are a type of protein that you need to kind of be paying attention to. So all living things are made up of chemical compounds, but it isn't just what life is. Chemistry is what life does. If you were just made up of a grouping of atoms all smushed together into a U-sized shape, you wouldn't be living, you wouldn't respond to stimuli, you wouldn't be able to reproduce, you wouldn't be able to do any of that thing, use energy, that kind of thing. We need to make sure that we are also moving. So that's what we're going to be kind of talking about today. The issue is chemical reactions are really slow. They're not terribly effective things. So we need to speed up these reactions. That's kind of what enzymes do. So here we have two different things, and we're going to be talking about them today. So things that are going into a reaction are known as reactants. A reactant is what goes into a reaction. So in this case, we've got our little red, I guess it's this way for y'all, this little red C. So this is going to be our reactant. When it goes into the reaction, it looks like this. After everything in the reaction happens, you get a product. So in this case, we've broken down our little C. So we broke down our C from product to reactant. Reactant, product. Sorry, that was confusing. Reactant is when it's together, product is after, and it's split apart into two pieces. So, energy is either absorbed or released whenever you have one of these reactions. So it's either absorbed or released whenever you have one of these reactions, and that's very important as we go through. So energy, things that release energy, so things that are breaking apart, generally don't need any help. They happen spontaneously and really, really quickly. You can kind of think about how easy it is for your whole life to fall apart if you're not really focused on it, but it takes a lot of energy to keep it together. So these are what we call spontaneous reactions. They just happen all on their own. So, energy sources. So we have to have some sorts of energy to carry out chemical reactions to stay alive. We talked about that was one of our 8 characteristics of life. It was that, reproduction, response to stimuli, several other ones. Make sure you know that for the test. Remember, characteristics of life is topic 14. But, enzymes. So plants get their energy by trapping and storing energy from the sun. They build molecules, and all of that energy is stuck between those bonds. We talked briefly, I think this class talked about the difference between potential and kinetic energy. Potential energy is the energy that's going to be stored in those bonds. So animals, we get our energy from eating things. So whenever you don't eat, you get kind of tired, lethargic, you don't feel good. That's why we get our energy from what we eat. And if we're not eating anything, don't have any energy. So humans release the energy needed to grow, breathe, think, dream, all of that through chemical reactions that occur when we metabolize or break down. So to metabolize is to break down. So if you have a good metabolism, you're good at breaking down food. If you have a bad metabolism, not so much. Digested food. So we need to talk about activation energy. Let me move myself over here. Where can I be? I can be down here. Okay. That way you can actually read it. So, activation energy. The peak of each graph, and you can kind of look down here, right here, and right here. So the peak of each graph represents the energy needed for the reaction to go forward. So if it doesn't have that initial boost of energy, nothing's going to happen. Needs that activation energy. And that's what it is. It's activation energy, the energy needed to start something. Think about how much more energy you need to get out of bed than you do to continue staying out of bed, which, by the way, is why you should never study or do any work or sort of that kind of thing in bed. It's just not going to work well. So the difference between the between the required energy and the energy of the reactants is called the activation energy. So you can see here we have a certain amount of energy that's trapped in our reactant. So this contains a certain amount of energy. The energy that's required, the energy way up here that's required for it actually to start, that's the activation energy. So from reactants up, that's going to be that. And you can see that even when we have an energy releasing reaction like this, breaking apart and becoming these, you still need some activation energy. It's just not nearly as much as you would need to build something. So this is kind of what you can think of. So without an enzyme, we have our cannon that's shooting the ball up the hill, and it just doesn't quite have enough energy to fully make it up. It's because this hill is too big. The activation energy is too much required. What enzymes do is they actually come in and they push that down. So we've taken this and we've actually pushed it down. Sorry, I tried to point at it and I can't actually do that in this. So we actually push down the required activation energy. You can see that that top hill is much bigger than the bottom one. So we've pushed down the activation energy required, and the cannon can shoot that red dot way over. So it's lowered the activation energy. So what that means is that enzymes speed up reactions. Let's say I need to move myself again. Okay. It's not much better, but it'll do. So enzymes. This is going to be one of our enzymes. So we can see it's got what's called an active site. It's got a deep fold right here. So that's going to be the active site. And it's kind of formed a pocket. You can put something inside of that. The molecule being affected is called the substrate. So in this case, we're using this as our substrate. So we've got our enzyme in yellow and our substrate in red. And on Tuesday when I get back, we'll actually be using these, and I'll give you guys these to work with. But for today, just kind of watch me do them. So we've got our enzyme and our substrate. Two different things. The enzyme never ever changes. The substrate does. So when the enzyme actually changes its fit to mold around that substrate. So let me grab another one. So this is our original enzyme, and we can see that it's changed shapes. It's now much bigger than it was over here. So it's changed shape to fit around our substrate. So it's called induced fit, or when something changes to fit something else. So induced fit is this. So it kind of moves the substrate into such a way that's more likely to react. So we can see that our substrate's kind of more squished together than it was originally. Kind of squishes together when it has to fit in there. So this is more likely to react when it's this shape. So if you look over here, we can see that our active site is a bunch of deep folds. It would fit around this substrate, just locks right in there, and then it changes the shape of the substrate. So when they're bound together like this is called the enzyme substrate complex. So the substrate binds to the active site. Remember, our active site was right here. So it binds to the active site, and they fit together. They have what are called complementary shapes. So when you complement something, you're kind of going together. So they're complementary shapes. They're really similar in size and shape. So I'm not going to use this enzyme to react with something like this, and we will talk about this later. But these two things don't fit together. They're not the same size or shape, whereas these are. So in order to fit, they have to be exactly the same. And we call this the lock and key model because you only have specific keys that can open specific locks. They have to match up or they're not going to work. So the enzyme has to specifically fit the substrate or it's not going to happen. So this is what it's going to kind of look like. So our substrates are going into the enzyme. They fit in there just like a lock and key. Some sort of reaction happens here in the middle, and then a product leaves. So if we're looking at this not on here, we've got our enzyme, and in this case we're building up a molecule. So we've got two different substrates. They're going into the enzyme together to form the enzyme substrate complex, and then chemistry happens, and you end up with a a substrate or a product that's been changed in our enzyme. Notice our enzyme has not changed from its original shape. This is exactly the same that it was going in. So the enzyme can be used again and again and again and again and again to turn more substrate into products. So very, very important there. Enzymes can push things together, they can separate them, but they're never going to change. So we call enzymes nature's catalyst. A catalyst is just something that makes it go faster. So they act as biological catalysts. They make things that need to happen. They act by lowering activation energy. We did talk about that, which has a dramatic effect on how fast things go. Everything would probably happen all on its own, but it's not going to happen with the speed that we need it to actually be effective. So carbon dioxide combines with water to produce carboxylic acid so slowly that carbon dioxide could actually build up and poison you. So not good, not where you want to be. Your blood contains an enzyme called carbonic anhydrase that speeds up this reaction by about 10 million times. So really, really much faster. It stops your blood from being poisoned. It's really important that you have it. So enzymes can be regulated in one of two ways. They can either have a competitive inhibitor or a non-competitive inhibitor. So competitive inhibitors are something that directly compete for the active site. Remember, the active site is right here. So if our substrate looks like this, we can have a competitive inhibitor, which is going to be our green one here, that can fit into this active site and stop the enzyme from working. Because if this is blocked, we can't fit our substrate in there. It's not going to happen. It's directly competing for something. It's using it instead, but this is not useful, so you can't actually do the useful thing with it. So this is a competitive inhibitor. And so that's bonding to the active site. So competitive inhibitors bond to the active site. Now, our other ones are non-competitive inhibitors. So non-competitive inhibitors are going to look nothing like it, and we can kind of see here on the back, I haven't talked about it yet, there's a site right here. Sorry, I point at it from this side. There's a site right there that's not the active site, because remember the active site's here. So it's not the active site, and this can just fit right in there. And so now this can't change shape like it was before. So there's not going to be a way that I can force this substrate in. It's changed the shape of my enzyme fundamentally. And remember, when we were talking about it, essentially what we've done is we've put glue in our lock. So our lock's not going to open to our key. So this doesn't work. So this is non-competitive because it's not directly competing. It's bonding somewhere else. And then our competitive is actually going to bond to the active site. So that's kind of the difference between competitive and non-competitive. I'm running short on time, so we're picking it up. But remember, you can always pause this. Feel free to take your time, go through things slowly. All right, so regulation of enzyme activity. Temperature, pH, concentration, regulatory molecules, these are all things that affect it. Think about how you work more effectively when it's not 10 million degrees outside. Enzymes do the same thing. They don't want to work when it's hot. They don't want to work when they're doused in acid. Generally, human body enzymes work best at 37 degrees Celsius, which is the temperature of your body. 98.6, 98.8 degrees Fahrenheit. That's what you guys are thinking in. Same basic thing. Depending on the job and the location of the enzyme, it's going to work best in a different pH. The ones in your stomach acid are obviously going to work at different pHs than the one in your blood. So you can kind of see that here. Okay, that is the last slide. Good. Not going to run out of time. I got exactly a minute left. So, pepsin works best at really low pHs, so it's got the highest enzymatic activity here. Amylase, which is in your spit, works best over here. And then arginase, which does something else, works best when it's more basic. So that's kind of enzymes. What I'd like you to do now is there is an virtual enzyme lab in the link right below this one that you need to go through and answer. You should have lab questions for that up on the lab bench up here, which is where I'm sitting. Please come grab one. I got 10 seconds left. So click on the next link, it should have more specific instructions. Okay, bye guys!